Right, so we are going to do about 3 things very quickly within 10 minutes. Within 10 minutes, and then I would have been done with you. So last Saturday, or let me say Thursday, we couldn't meet for the variant calling. And remember, we're having some assignment on the read alignment, and we couldn't do that one as well. So uh, we are going to be looking at read alignment, sequence mapping, and then variant calling. We also try to look at different types of variant calling, where we look at indels and then we look at SNPs. And we would have done our variant calling, and then we can go into that and actually segregate which of the variants were SNPs and which of them were actually indels. If you remember in our discussion, we had spoken about different types of variant calling. It could be germline, it could be mutation, or it could be somatic mutations. And that's why we have the indels and whatsoever. So I was thinking that you guys were going to do it. Fortunately, you couldn't do so. I'm going to share. I'm already sharing my screen now. So I'm going to pull up the order of how you're going to do it. So we have read alignment. So this is the order. I just structured this particular small graph for you to see. So the process is here. I'm going to share this particular document with you. The links and everything are there. So where I'm highlighting right now, that's the process. First of all, go to your Linux, like I am here on my Linux right now, and create a directory. When you create that particular directory, you can simply go to and then you cd into that particular directory. So I call mine what is the variant calling. Then into that particular directory, then you go to this particular link. It's a website where you can get the paper where I got the data from. So on that particular paper, you're going to find this particular uh, accessing number, and then you can use the accessing number and follow this link I have here to NCBI SRA link website. Then when you go there, then you put the accessing number, and then it's going to take you to this particular page where you can have this is the link to downloading the read one, and this is also the link for downloading read two. It's a paired end read, so you should get to know that. So to be able to download it direct into the directory you have created, now use widget or wget plus this particular link. So this is how you're going to do it. For instance, if I were having this, I'm going to go back quickly. So I have just created a directory called var calling. So I'm going to cd into that particular directory, sorry. Then I will just cd and say maybe var calling. Then I'm going to go into that. And then I will now use something like this, wget, then you add the link to it. When you do that, it will download the read straight into the directory so that you don't waste your time and then redirecting. So when you've done that for, let's say, read one, you do the same for read two, then you come and download the reference genome. The reference genome, we are talking about a bacteria. I'm no longer dealing with the human one. I was going to use both type for the human genome. Now I'm using a bacteria. The reads are from a bacteria, so I'm going to use the reference genome for this, the Mycobacterium tuberculosis, which is the AGY99. So go to this particular paper, the link is here, and then you can download the reference genome using the same analogy I have just narrated for downloading the reads. Now, if you have done that, then you would have all your reference genome and that of the reads you want to align into the same directory. And you're already working in that particular directory. You are also going to use the same widget plus the link to that particular reference genome. And then you just click enter. It will download everything into that particular directory. Now, we have that ready. This is the second stage we are going to do. Now, when we have that, they come in the form of zip files. So instead of unzipping them, we're not going to unzip them. We're just going to go straight to check whether the quality is good or not. So we come to use FASTQ to actually check the quality. And when we do the FASTQ, this is the code. And in all of them, you can see that I put code, and that's the code I'm just underlining here. You write FASTQ plus the name of the read. So if you had downloaded the read one and the read two, following the direction I've given to you, you will see the read one, it will end with GZ, or it will be R1. The read 2 is R2. You will see that over there. I can just show it that quickly. If I come to my downloads here, you can see the file, the folder here. So you can see this is the read 1. You can see the read 1, it is being highlighted here where I have marked, and then the read 2 is also here. So when you do the quality checks using the FASTQ, an HTML file will come, and then that's where you can actually open it to see how, you can see that this particular read 1 is very poor. And to be able to interpret that, I'm going to leave that for you to also try your possible best. This time around, please, I beg you, everybody should try and then also look at how to interpret this. But obviously, if you are seeing the red portion is no, the yellow is warning, and the green is actually good. So we are going to, if we have that quickly, what that means is that we need to do some kind of trimming and, of course, filtering. Get to know the difference between filtering. Filtering, you are removing the entire read. The read is not nice. So you are going to filter it out. For trimming, you are going to remove some bases. It means some few bases are not good, and you are going to trim that off. That's the difference. So we're coming back to the same process. Now we've done our quality checks. Then the next thing is to trim. And I'm going to use this particular, I'm going to use this particular tool called Sickle. If you don't have Sickle, go to Anaconda website and then install your Sickle. Now, when you have your Sickle installed, this is the code that you can use to do your trimming. And now, after you have done the trimming, obviously, you are going to have these 4 files coming out. You have the trimmed already, out here. Then the next thing to do is to do indexing. Please try and know why we do indexing when we are doing read mapping or sequence alignment. Now, indexing is like you have a lot of students and they all have index numbers. The index numbers, actually identify each particular student with every feature that the student has. So if you want to know me, you use my index number and you can get to me. The same way we are talking about indexing. So the indexing is to simply identify every single base and the features, or every single code within the genome, or every single gene within the genome and all the features. So it will identify each of them with a particular given number as a reference, which we call indexing. So when you do that, it makes read alignment or mapping quite easy. So we do the indexing using this particular code. So this BWA means barrel wheel alignment. It's actually a tool which we use in doing indexing. We're also going to use the same tool for the alignment. So after we've done the indexing, in fact, let me say this, we don't index reads, we index reference genome. Okay, we use the barrel wheel alignment tool to do that. And we come to do the mapping right now. So we're mapping the reads to the reference genome. And you use this particular code I have given here, and you are able to do that. After the mapping, you are going to generate an output file, and which will be a SAM file. The SAM file is quite heavy. So what do? We convert it into a BAM file. The BAM means binary alignment mapping. So you convert that to just to reduce the size. And I've decided to do a comparison for you to see the SAM file and the BAM file, and then which one is heavier. You can see that the SAM file here, to be able to do that comparison, use this code, du-sh, and then you add the file name to it. It's going to give you what you can see here. It means the SAM file was 296 megabytes. But when we converted it to BAM file, the BAM file is 92 megabytes. See that one is quite smaller. The next thing to do is to do sorting. We need to sort the BAM file that we have right now. And then we can use these SAM tools. If you don't have SAM tools, you need to install it using Anaconda. Andrews already showed all of you how to do installation. So you should know that quickly. So you use this particular code, you can sort your BAM file out. Now it's time to do the mapping. So after we have done that and we have done the mapping, we need to come out with the statistics of the mapping to know that, okay, how much was the reads mapped, what percentage was mapped correctly, how many of the reads were mapped, how many percent or what percentage failed, and what happened. So all the statistics will be there. So you use this particular code, you use some tools to be able to do that, and you flag the statistics, and then you output it as a sorted BAM file. Then This is the output file of the statistics. Of course, if you wanted to write a report, you can actually copy this into, you can save it using this particular code I have down here, where I've highlighted. It will be saved into a mapping statistic.txt, and then it will be there. So we can use that to be able to, I don't want to do this repetition, because when you use this video with this document I'm going to send to you right now, you should be able to do exactly what I've done, and you should be able to get exactly what I got, because I've already run all of them and they've worked perfectly, and that's why I'm showing the results now. So after we have the mapping statistics saved into this file, then it means we can write our report against it. So now we need to know the coverage. How much was the coverage of this particular mapping that we did? How much was the genome covered? So we use the MPyLab tool, which is also one of the tools that come with the BCF tools. I think at this point, let me emphasize that if you are working within an Anaconda environment, BCF tools doesn't work there. You need to move out of the Anaconda environment to be able to run this code. So I tried it and it didn't work. And the reason could be that I've already got some tools in the Anaconda environment which is contra, or which is not incompatible with the BCF tools. So when you run and it doesn't work, move out of this Anaconda environment. To do that, just say Conda deactivate, and you move out of it. Later on, if you want, you can say Conda activate. You go back to your Conda environment. Then when you run this, it's going to give you the MPyLab. Because if you don't do this, you won't be able to do your variant calling. Now, it's not time to do the variant calling. So here, we come to run our variant calling using this BCF tools. And let me explain this code. The BCF tools is what is going to do the actual call of the variant. Now we know what variant is all about, the difference that we're going to see between the genome, the reference genome, and that of the RIS. Those are the variants, those are the variations. But when you are dealing with a human genome, that's a diploid. So you put 1. Here we are dealing with a bacterial genome. So we put the ploidy level, we put 1, which means it's not a diploid. That's the difference. That's why you see 1 here. And then you add the variance or the file to it. And then you are going to save it as a raw BCF file. After that, when we run it, it will call all the variants. Now, how do we know how many variants were identified? How many? Even though it was in the text file, but we can actually grep and count them. So you use this particular code to grep, and then you count them. As we can see, we have 32,828,387 variants that were called. But which of them were index, which of them were SNPs, and which of them, also fell into a different category of the variants. Variance is not only one. So to find out those variants that were SNPs, we use this particular code we have here. And then we can actually count out of the total 32,000 variants we have, 30,000 of them were SNPs. It's very simple to report on this. And then how many of them were index? About 1,525 were index. We can actually visualize this thing using VCF manipulation tools. But that is not within the scope of this tutorial. If we find that necessary, I can do that as well. So, but we also need to check something very important. Of all the variants we have called, what are the SNPs, the index, and whatsoever? Now we know where in the genome were these variants identified, if they were SNPs, where specifically. So we need to find out the positions within the genome where the variants were called. So to do that, you run this BCF code here, and of course, you can actually identify the positions. And I've decided the positions are quite many. You can't display it on your screen. So you decide to save it into this particular file called position.txt, and then you can use something like a head. You can just use your head, let's say head, and then you add that particular file, let's say pos.txt, ts, and then it's going to give you the first 6 lines of those positions that the variants were called. And of course, For instance, if I come here, I'm now on my Linux environment. I could just say head, and then I'll have my POS. Sorry about that. POS dot. If I just do tap, it should complete it like this. So you can see that there are just too many. You can't just. So these are the positions on the genome where the variants were called. And as I'm saying, I've already run them, and that is why I don't want to waste everybody's time. So that's about it for the three things we're supposed to do: read alignment, sequence mapping, variant calling, and then counting the variants and identify different variants on a genome using a bacteria reference genome, AGY, and that of the two REEs, which we found at the Mycobacterium tuberculosis or Ostrans. That's about it for today. Tomorrow we'll be doing something a little bit different. If you have any question, you can put it on our WhatsApp page, and I'll try and see if I can get to that. Thanks, all of you. I'll see you some other time.
Now, be a very great nature.